Again, good evening. My name is Karen Slavinsky, and I'm very pleased to introduce Stephen Russell, President and founder of the Hoosier Mushroom Society. Now, I paused on Hoosier because I had to look it up. Hoosier is the pet name, the nickname of people who are proud Indianians, people from Indiana. So Stephen is from Indiana and educated and is still getting an education there. He founded the society in 2009, and by 2015, he was leading his society with the use of Sanger sequencing in identifying by DNA identification fungus in Indiana. Currently, he is using Oxford nanopore technologies, and nanopore is showing to be faster and less expensive than any other DNA sequencing technology. Of course, still requires DNA extraction, amplification, tagging of samples, and pooling for sequencing, and then, of course, the important step, the analysis of the DNA. Stephen has developed the nanopore barcode protocols and has shared these in the public domain. And so if you Google him, you will find them. And he has shared them with our society, but they are mind-boggling. But they need us, because citizens who forage are extremely important, because this protocol requires large numbers of fresh specimens. These provide the best sequencing outcomes and very useful for the protocols, and actually very useful for involving many citizens. So why is the Hoosier Mushroom Society interested in identifying mushrooms using DNA? Well, there are many reasons as to why. One, of course, is that it is a very conclusive and useful method for identifying, but it is also very important in documenting the distribution of fungus in Indiana. And of course, if we adopt it in Alberta. So Hoosier Mushroom Society is not only what Stephen is. He is a young father, but he is also a PhD candidate in the Department of Botany and Plant Pathology, studying the biodiversity of fungus, some fungi, and some specific fungi which act as a disease agent on American chestnut, and of course, among other things. Stephen is presenting tonight because he, like the Hoosier Mushroom Society, because we, like the Hoosier Mushroom Society, are interested in DNA barcoding of macrofungi in Alberta. So let us welcome Stephen tonight for 1 hour to share his expertise. I had to tell you about the mind-boggling email. 1 hour does not do justice to his protocols, but it is actually a great little entry point. So welcome, Stephen.
Yeah, many thanks, Karen. So I guess I would start out by commenting on a couple of things that were mentioned. So Hoosier is only somebody from Indiana, so nobody really knows where that word originated from to this point. There are multiple competing theories about where it may have originated from, but that's all it means is Hoosier is somebody from Indiana. One of our largest state schools is actually named the Indiana Hoosiers. You may have seen them on television. One other thing I guess I would mention from the introduction, one thing once you get into DNA research, you'll quickly realize that even with DNA, it's not really conclusive for many species for many different reasons. And so there is actually still quite a bit of subjectivity in the entire process when it comes to actually analyzing the results that you get from DNA. And so that's something I would kind of ask you to keep in the back of your mind. We'll talk through a number of things with DNA sequencing. That won't be the only thing the talk is about tonight, but it will be kind of the bulk of the presentation. Let me go ahead and share my screen. The host has disabled screen sharing, so I need permission before I do that.
Yes, just 1 second.
And so it was mentioned that the Oxford nanopore protocols are substantive, and they are quite substantive, especially if you're new to the entire process. I think there's the protocol is actually composed of maybe 7 different protocols. And so there is a lot that goes into the process. But if you've done Sanger sequencing in the past, I think you would find that it's at least as easy, if not easier, than Sanger overall. Let me see if I can share again.
I made you a host, so you should be able to.
And I can.
Good stuff.
And if I can minimize, what the heck? Do it from up here. So as was mentioned, my name is Steve Russell, and I'm with the Hoosier Mushroom Society. The bulk of what we're going to talk about is sequencing in some form. But one of the things that I do want to get across is kind of our approach to DNA sequencing. And I think that's what we'll start with today. And really, we started with the underlying goal of knowing all the mushrooms that exist within the state of Indiana. And so if you think about it, that's obviously a pretty lofty goal. Indiana's a somewhat large state. If you were to drive, from the north to south, it easily takes over 4 hours to get from the top to the bottom. So it is a pretty large land area. And to this point, I would say still to this point, there's really no region of that size that has any type of rational understanding about the totality of mushrooms that actually macrofungi, mushroom forming fungi that actually exist within a land area that scale. But that was our underlying initial thought process and goal of how do we even begin to approach that kind of question. And so one way to think about surveying biodiversity or beginning to answer that question is by thinking about the different scales of biodiversity that are out there. And so alpha diversity, beta diversity, and gamma diversity are 3 that are commonly mentioned within the scientific literature. Let me just briefly go through them to kind of give you an understanding about what they mean. So over on the right, I still can't minimize this. There we go. Over on the right in this image, you see an aerial photograph of several of my research plots, not just mine, but they're part of a larger hardwood ecosystem experiment. So there's 11 plots, and you're looking at something probably about on the order of 10 miles from top to the bottom. And if you're thinking about alpha diversity, that could be thought of as the diversity of one of these plots. So if you were to go to one of these plots, survey all of the macrofungi or anything else, whether it's trees or plants or animals, alpha diversity could be thought of as the diversity that's present on one of these plots. If you take it up a level, beta diversity could be thought of as the difference in biodiversity between 2 plots. So you have alpha diversity of plot 1, alpha diversity of plot 2. Beta diversity would actually be the difference in the biodiversity between these plots. Going up one more level is gamma diversity, and that can be thought of as regional biodiversity. So if you were to look at the biodiversity of all of these plots over a 10-mile radius, 10-mile diameter, then that could be thought of as gamma diversity. You could also think about it as regional diversity. And that's really more or less where the scientific literature goes to. They don't, just because of the difficulties involved in assessing biodiversity over these large geographic areas, you don't really get too much going into the step above, which is epsilon diversity. And that could be thought of as biodiversity of something on the order of 1 million hectares. I think Indiana is about 10 million hectares. So something on the order of a state. What's the biodiversity of an entire statewide region? And that's epsilon diversity. So that's one of the largest scales of diversity. And that's really what I'm interested in studying for most of my research. One of the primary ways we do this is by hosting events online twice a year. So typically in the summer and the fall, we hold events that we call online forays. And there's a paper online, a preprint that you can check out if you're interested in this, but this has been by far the most effective way for us to begin really capturing biodiversity on these really large scales. And so we've been doing this, I think the first one we actually held was in 2017, I want to say, and they keep on getting larger and larger every year. But for any one of these events recently, we get well over 3,000 observations. And the most important part for me in this is people don't just post their observations on iNaturalist. They will actually save the specimen, dry it out, post their images on iNaturalist, but then mail me in the specimens. And so for what we're really attempting to achieve, we want physical specimens associated with all of our iNaturalist observations that are occurring. So as an example, for our latest summer foray for 2022, we had about 3,400 observations. And people actually sent me about 1,700. I'm still working through them a little bit. I don't have a hard count, but I think we've sequenced somewhere on the order of 1,500 to 1,700 specimens from that single event to this point. These events are week long. So we pick a week during the summer. It goes from like a Friday to a Friday or a Friday to a Sunday. And there's a whole bunch of benefits for this type of model relative to the way traditional forays have worked. So with a traditional foray, you might pick one specific location, have everybody drive to that location, meet up at the specific time. And many people are often limited by, their work duties, their family needs, just the cost of actually getting in a car and driving anymore can be quite substantial, as well as time commitment. So as I mentioned, let's say we wanted to hold a foray in southern Indiana. It would take the people in northern Indiana about 4 hours to get down to that event. And so by holding these events online, we allow people to get out in their own locality. They can go to their own local park. They could go out to their own property if they want. And they can do all their collecting. They can take all of their images, post them to our individual project for the event. They can do it on their own time throughout the week. So that way, if they have to work on a Saturday part of the day, they can still be able to participate at a really high level if they're so inclined. And so this has really been one of the top methods for us to begin systematically capturing as much biodiversity as possible. So many of you are probably also aware that, let's say it hasn't rained in one portion of the state during the summer. Well, that's fine, because there's typically with these larger land areas, there's going to be somewhere in the state that really has mushrooms fruiting really well at any one point in time. And so we aren't limited by the fact of, if we were to pick a single location at a single time within the year, it may be really dry and there may be no mushrooms out, and then we don't capture very much biodiversity. But by making these events distributed across these really large land scales and allow people to contribute at their own location at their own time, that really allows us to capture as much biodiversity as may be out at a given moment. And so I encourage you, especially if you're thinking about starting a DNA sequencing project. We'll talk about it in a little bit once we get to nanopore sequencing. We're really limited by the number of specimens that are available anymore. And so a single nanopore run, we do about 1,000 specimens at a time. And most people have never collected, and most organizations, most mycological societies have never collected 1,000 specimens as a society, in the history of the society. It takes a lot of work and a lot of effort, a lot of organization to be able to do that. But that's really, if you're trying to make the most of a sequencing project and try to capture all of the biodiversity that's there, if you start with that original premise of we want to know everything, the number of modern collections that are available is often going to be the limiting factor. And the primary way to overcome this that we've found and that we've been really successful with, I would say, is through these online forays. Two week-long forays a year. We do have smaller events that we'll do as well throughout the year. But if you, I guess one other thing I would mention on this, if you look at this top portion, Indiana Fungi 2022, all of our top collectors will be collecting things all throughout the year. And so it's not, the collections aren't limited to these forays. The forays are just kind of a way for us to have a fun, exciting, event style structure to allow everybody to come in all at once and to look at everything everybody's finding all across the state. And so it ends up being a really fun and interesting event for us, but it's not limited to those weeks throughout the year. Through our yearly projects, Indiana Fungi 22 is the latest one. People can collect at any time and then mail their specimens that they're finding all throughout the year as well. And as I mentioned, that one of the advantages of this is it allows us to get collections from a really broad geographic area. And so this is our current map of iNaturalist observations from Indiana, where voucher specimens have been retained. And so as you can see, we're really trying to make an effort to cover the entire landmass of the state of Indiana. And so understanding what occurs, where it occurs, and when it occurs is really foundational knowledge for most further research within mycology. And so that's often a question I get quite frequently is why are you even doing this? And the best answer I found to this point is that understanding what occurs, where it occurs, and when it occurs is really foundational knowledge for many other areas of many further lines of scientific inquiry in various other areas, whether it's fungal ecology, looking more at the structure and the function of these things, or whether you're looking to examine climate change, how the species distributions are shifting over the course of time. It's very important in many applied mycology topics. A lot of people are into medicinal mushrooms as an example. And so understanding what species even exists is foundational knowledge for most claims about medicinal mushrooms. And just, I guess as a side note, that's actually been a substantive issue within the research for medicinal mushrooms, is that people would often think they're doing research on a given species, and it's later determined that all of their scientific research was based on a different species. Just like with Ganoderma lucidum, there's a whole bunch of lucidum type things that are out there worldwide, and whether or not you have the real lucidum can affect, the amount of specific chemicals that are produced within a mushroom fruit body. As an example, if you're into studying, the benefits of Ganoderma lucidum, you better make sure you have Ganoderma lucidum to begin with. And so that's kind of what I'm getting at, is that it's really foundational knowledge for every other line of scientific inquiry that you may be interested in with mushrooms. And so understanding what occurs, where it occurs, and when it occurs is kind of the underlying rationale for a lot of what we do. And so since 2018, we've been surveying Indiana somewhat substantively. We've really started this thought process in 2015, but it wasn't until 2018 when we really got our act together to start doing it in a large scale, organized way. That's kind of when we started doing our online forays and having lots of people participate in those events. We're currently, I believe, over around 15,000 sequence specimens that we've been able to collect and retain and sequence since that time. We'll probably be over 16,000 by the end of the year. And we're somewhere in the ballpark of 2,500 species that have been recorded. And so this is an ongoing effort. It keeps growing every year. And our goal is really to get to the end. And so that kind of leads you to another question, is about how would you even know that you're close to the end of a project like this, or a thought process like this? And there is actually a way to do that, to start to make an assessment of how much more collecting do we have to do still in order to attain a reasonably comprehensive survey of all of the mushrooms that exist within our state. And so this is something called a species accumulation curve. And across the bottom is the number of collections, and across the y-axis is the number of species that are involved. And there's a whole bunch of different ways you could look at this chart and a bunch of different types of analysis that you could do with a simple chart like this. But the one I'm most interested in is that for the last 1,000 collections, we're still averaging. And so this, I guess one qualifier with this chart, this is the number that I've been able to validate to this point. It's somewhere over 7,000. Just within the last, this year, we went from about 10,000 to 15,000 specimens with nanopore sequencing. And so it takes a long time to actually go through and validate these things and to delimit the species that you believe to be novel or otherwise interesting. But for the last 1,000 collections, we're still averaging about 1 new species for every 5 collections that we make. And so if you think about this, we're not, especially during this time, we weren't just sampling everything that's out there all the time, right? We probably have 40 or more sequenced specimens of turkey tails to this point. And so we're no longer accepting new specimens or retaining. People can still send them in. And I guess that's one point I can make with those online forays. Everybody understands that they would much rather have someone who's skilled make an assessment of whether or not something's important or not. And so as people collect more and more, they might photograph a turkey tail now and they wouldn't collect it. But it's fine for a new collector if they aren't sure what they're doing to go ahead and just collect everything and send it in. Once they send it in, once they mail it to us, then we'll actually do a triage on the specimens and make an assessment of what's important or not. And that's one of the things. So as time's progressed, early on, we were sequencing all the turkey tails that would come in just to try to get an assessment as to whether or not we have any novel species of turkey tails, anything that's new or interesting within the turkey tail complex. But as time progresses, we've been getting more and more stringent in terms of what we save, because what was really interesting to us several years ago, it's not really interesting to us any longer. Just because we've seen that species so many times now, we have a whole bunch of sequenced specimens of it. We know that it's not a group, and that there's probably not much interesting going on there. And you can still always get surprised, right? So you might look at 20 specimens of a given species, and then the 21st one appears to be novel. And that definitely happens from time to time. And so that's actually one of the reasons why you kind of want to make the collecting effort broad-based, and that's especially true even for things that you think you know. Because as we'll talk about in a minute, quite often things you think you know, you don't, nobody probably knows quite as well as you would think. But what we're really looking for with this chart is to say, we know we're nearing the end of our collecting effort, that for if we took our latest 1,000 collections and we only had, a new species for every 20 collections made, or a new species for every 100 collections that we make. And as you start getting closer to that number, if it's taking 100 new collections rather than 5 to find a new species, this chart would start to plane out. So it would almost be a straight horizontal line at that point. And so that's really what we're trying to do. It's called bending the species accumulation curve, and really trying to get cause this accumulation of species to go horizontal, to not have many new species, to collect a whole bunch of collections, and to not have many new species coming in. And so over the course of time, you can see that for the first 1,000 collections, if you were to look at the slope of that line, it was a much different slope than it is now, but we still aren't seeing much of a bend to this curve in terms of anything in the horizontal direction. And so we still have a long ways to go. But for individual groups of mushrooms, we are starting to see more of a bend. And so this is a look at Amanitas, and I'm still dealing with the issue of, let's see, there we go. I think we're somewhere around 1,000 collections of Amanitas to this point, somewhere on the order of 160 to 170 species. And for Amanitas, we're averaging 1 new species for every 17 collections that we make. And so as time progresses, we're still collecting lots and lots of amanitas quite regularly, but our rate of accumulation of things that we believe to be novel to our data set is really slowing down relative to a lot of other things. If you were to look at things like Russula or Anasabi, we are still seeing no bend in the species accumulation curve for many things. And that's primarily because amanitas have always been a focus for us. And so people are regularly collecting lots of amanitas, you know, especially for most people in the club, they're large and showy and interesting in a lot of ways. And so we get a whole lot of collections of amanitas relative to most other groups. And that's probably why we're seeing more of a bend in the species accumulation curve here relative to some of the other groups that are out there. I would also mention that if anyone has a question, I see there's nothing in the chat at the moment, but feel free to go ahead and pop something in the chat if you have a question as we're going through this. And so this is a long way of getting to our ultimate goal of knowing all of the macrofungi that exist in Indiana, being able to quantify how many species of macrofungi do actually exist within our state, and knowing how much work we have left to go. So if I were to give an estimate on how many more collections that we need, if we're currently at about 15,000, I would think my current estimate is probably about 40,000 collections to have a reasonably comprehensive understanding. I believe that's ultimately where we're going to need to be before our project can really die down in any significant way. When I first started this, I was thinking 10,000 collections may do it, and that was our initial goal. But we're well over 10,000 collections now, and there was really very little sign that we were starting to finish up in terms of documenting all of the biodiversity that's out there. So I mentioned this a little bit, but let's dig into kind of the underlying question that's a part of that goal. And that's, how can we document the most biodiversity at the broadest scale in the shortest timeframes at the lowest cost with the highest accuracy? And all of these goals are somewhat, you know, they can be conflicting with each other, and you might gain a little bit of accuracy and lose a little bit of cost or something like that. But all of these factors are really important to us to do it at a large and broad scale, as quickly as possible, as cheaply as possible, with the highest accuracy. And ultimately, what we came to balance all of these competing goals was the citizen science collaborations that we talked about, primarily with organizations of our members, especially doing the online forays, and DNA barcoding of the ITS region. And we'll talk about that here momentarily. But are there other options? Is another question. You know, is there any other way to approach this type of goal that might be just as effective? So traditionally, most mycological societies have used species lists in terms of aggregating information over the course of time about the mushrooms that exist in their area. And so I'm not sure, I should have probably looked this up before I got on here, but I believe you guys have Pluteus vermellii out there, what you believe to be Pluteus vermellii out there on the West Coast. And if you start to think about, this was a really well-known species. It's really easy to identify. So a small brown Pluteus growing from wood, free gills, pink spores, with a brown cap and a bright yellow stem. Everybody thinks they know this species, and it's probably recorded, if it's in your area, on many species lists going back throughout time. But it leads to the problem that we refer to as cryptic species, because everything in the Pluteus vermellii group in North America is not actually Pluteus vermellii. And this is, you guys have probably heard about this type of scenario with other species. I chose Pluteus vermellii here to illustrate it in this case, because it was just published about maybe a few months ago, over the summer. This work was published primarily from Alfredo Justo, who works on Pluteus a lot out of Canada, out of Eastern Canada. and dealing with a lot of species from all of North America. But Pluteus vermelii is a strictly Asian, Eurasian species, so Asia and Europe. And it actually consists of 4 different lineages here in North America. And so in Indiana, we had been calling them Pluteus vermelii 1, vermelii 2, and vermelii 3. So we knew we had at least 3 species based on the DNA. And none of them appeared to match the standard vermelii. And so this is what we mean by cryptic species, is that there are multiple species that are actually hiding under a single name. They're cryptic in this case. There is actually a 4th species that we don't have from Indiana yet, but we might find it at some point in time. But there's actually 4 different vermelii species in North America. And so we started this by not talking about cryptic species, but about species lists. And so if you think about a species list, and if that's your primary means of documenting something, your finds on a given foray, and everyone thinks they know Pluteus vermelii, and they did up until this summer, everyone would think, oh, this is an easy species to identify. We don't need to save any specimens. We can just write down the name and be good to go. But now if you were interested in, now with the additional knowledge that vermelii is actually 4 different species, if you wanted to go back and revise your species list, it's impossible. There's no way of knowing which of these Pluteus vermelii species, whether it's Parvosporus, Austrofulvus, Valingae, or Fulvibadius, that you actually have in your own locality, because the only thing that was documented was Pluteus vermelii, which we now know does not occur in North America. And so the traditional use of species lists, and I'm not trying to knock species lists, I still use species lists all the time for many purposes. But if all you have is that list of these names that are being saved, long term, it's not going to have very much lasting scientific value, because this type of thing is going to occur for, I would say, an outright majority of the species that you think you know in your area. In Indiana, we've been able to, we have pretty good data now on the number of things that we thought that we knew that would fall into this type of bucket where there's cryptic species or other things going on. And it's a very, very substantial number. And it involves many, many, quote unquote, field guide species that everybody thinks they know. And so the lasting benefit of the way mushrooms have traditionally been documented by North American clubs, I would say, 50 years from now, people are going to look back at the old species lists and there's just going to be no efficacy for them at all, right? They aren't going to be able to do anything with the species list because there's no, because we would now know that those names just are no longer in play. The way to give your species list lasting scientific value is to actually document the specimens that are occurring with each of the things that you're putting on the species list. Because then there's the opportunity to go back in the future and say, oh, hey, we documented this species at this location at this time. We now know the species doesn't occur in North America, but we have a specimen that we can go back and get more information for and actually update the name. And so that's one of the top things with this talk that I would encourage you to do, is to begin a thought process of systematically saving specimens. Even if you aren't DNA sequencing everything that you find yet, I can promise you that DNA sequencing is going to be cheap enough and available enough that here in a number of years, you're going to be able to go back into your herbarium and all of the specimens that have been saved, even if it's something like 10,000 specimens, here in a few short years, you would be able to go back in and have DNA results from all of those specimens within a year or something like that. And that's going to allow your species list to have that lasting scientific value because they're anchored to a physical specimen that you're able to go back and to examine as new information is available among these groups. And so just to reiterate, saving specimens in concert with species lists is really going to be a necessity going forward. And as I mentioned, there's lots of cryptic biodiversity in many places. So these are some common species that occur in Indiana. You guys probably use these names for a number of things out west as well. Something like Scutellaria scutellata, the little eyelash cups. That was kind of a funny story because we started saying, okay, we have 2 species, 3 species, 4 species. Over the years, we kept on getting, a new species, every year or something initially, but most people weren't collecting them. And most people, they weren't saving them and sending them in probably because it was it's a field guide species. Everyone thinks they could identify pretty reliably. Well, once we started looking at these eyelash cups this last year, the number just started exploding. So I put that on our focus list for the year of things that we need to get more information on, to start collecting everything you find. Anytime you see a Scutellaria, to collect it. And now that after we've been doing that for a year, maybe a year and a half now, our numbers went up to 13 or more Scutellaria species within our state. And this is backed up by other research that suggests there is a large diversity of Scutellaria. But I mean, just a few years ago, I'd never even really thought about that genus, and just kind of thought about it as one of those field guide species that, there's a species, we'll just slap this name on it. There can't be that many of them. Well, there's a whole bunch of these things. And that's really what you start to find with many different groups, and many of the most common field guide species. They're actually composites of many species, and that you can quite often sort out with some reliability based on morphology and microscopy. But I just kind of picked these names almost at random, as I was working on this presentation, what popped up that I happened to be looking at. And you can see, like for Leucogerchus rubrotinctus, 6 or more species just in Indiana. That's not including any of the West Coast things that go under the rubrotinctus name. We have 5 or more species of Leucogerchus that stain blue. And so there's just lots and lots of things out there to discover. And really these broad-based collecting efforts and saving the specimens is vital to be able to sort out what's actually occurring with this biodiversity in many of these different groups. So another way, so thinking back to what we initially started talking about in this section, is, are there other methods that we can use other than the broad, other than citizen science collaborations and DNA barcoding? Another thing that people have done historically is look at herbarium specimens. And that's something that the professionals still do quite a bit. They're really interested in herbaria and saving these things. And this is, an herbarium is ultimately where dried collections would end up that I'm talking about. So really all of our collections, currently I retain most of them locally outside of an institution, but ultimately they will all end up in an institution for permanent and long-term storage. But another thing you could do to kind of assess how much biodiversity occurs within an area is to say what's ever been collected in the past. And so that was actually done for Indiana. Scott Bates, he lived in Northwest Indiana. I believe he moved out of Indiana at this point. But he compiled a checklist based on all of the herbarium specimens that have ever been documented from the state of Indiana, and came up, I think they called it a proto checklist of Indiana species. And so historically, if we look at something like Agaricus, one of the most well-studied groups, I would say. They're very large, they're showy, people often see them, and they've received quite a bit of attention in the past, particularly with the Agaricus of North America book by Rick Kerrigan. Historically, 13 species of Agaricus had been applied to Indiana. And so up to this point, I think we had 168 specimens of Agaricus, and we got DNA from about 158 of them. And if we were to compare our results to what's on that list, the underlying question here is, how relative would a list like this, a species list generated from herbarium specimens, be based on a modern understanding of the genus? And it's not advancing. There we go. only 5 of the 13 names that had been historically applied to herbarium specimens are still being utilized. We had to add 17 new names to the list, and we have 7 species that are currently undescribed, even with the work of Rick Kerrigan. Above, and so if you look over here to the right, the ASM 13217 Kerrigan and the CF approximans, he actually included several of our undescribed species within his book, with these temporary code provisional names. And so these were things that he knew existed and, thought they were likely to be a unique species, and included them in his book, but he didn't go through the process of formally describing them. But the overall point here is, I guess, several things. Utilizing a almost generic name export from professional herbaria isn't really a ideal way to assess the biodiversity for a given region for a bunch of reasons. One being, more than half of the names that have been applied to the species should not have been applied, or are no longer being applied in a modern context. And then it's highly incomplete. We more than doubled the number of names that were originally on the list. So we started with 13, we had to add 17. And then on top of that, only 5 out of those original 13 are still being utilized with what we're doing today. And so I guess another thing I could note here is in the top right, you'll see Agaricus species IN 012345, and that's how we designate our things that do not have adequate reference data for us to compare to. So we'll talk about that here in a second, I guess. But that's often how we delimit things that we believe either to be novel or that otherwise need more work to be done. But the same type of process works very well for most taxonomic groups, not just Agaricus. If you were to look at Amanitas of Indiana, there were 37 species in the checklist that had been applied to collections historically in herbaria. Out of those 37, only 17 names are current, and we have about 150 total species. And with the work of Rod Tullis, we believe probably about 2/3 of our species of Amanitas within Indiana are undescribed. For Inocebi, we had 17 species in the checklist. There was really, really short because we're well over, I think, 160 species now of Inocebi within Indiana. We always knew these groups in particular were going to be among the most biodiverse, just based on looking at the DNA in the environment. So it's called eDNA. You can actually take small amounts of soil from many different locations, look at the DNA in the soil. Don't even need to have the fruit body and begin to get some preliminary assessments of how many species are likely to exist just based on the DNA of the mushrooms in the soil. And so with things like Inocebi and Ruschula, we had an inkling and we had an understanding that those were going to be among the most biodiverse in our state. But for Ruschula, we're at 88. We're at over 170 total species, and that 88 originating in herbaria was actually pretty surprising to me when I first saw that number, that people would actually put 88 different names on Ruschulas from Indiana. But we're well over 170 total species of Ruschula now, based on about 1,000 collections. And that's another group. All of these groups that outlined here, we've made substantial efforts to collect. There's people in Indiana, they'll just go out and collect every Ruschula that they find on a given walk. Not every single specimen, but every new location that they find something that looks different than they saw before. And that's really the only way that we've been able to begin to get a handle on the biodiversity. I think we have, someone tallied it up in our Facebook group last week. I believe, I want to say 50 to 60 of these Ruschulas are red, red capped. And so just if you look at the biodiversity of red-capped Ruschulas within our state, that alone is quite a substantial number. And there is a couple questions in the chat. Is it necessary to save the whole specimen each time? And so that's actually something that we've evolved with over time. If you take a traditional thought process with herbaria, they really want nice collections. They want samples from when they're young to when they're old, dried specimens from when they're young to they're old. They want multiple specimens. And that's something that we've kind of fundamentally changed our philosophy on over the course of time for a bunch of reasons. One of the main ones is just space. If you're trying to save 10, 15,000 specimens, or you're trying to mail specimens, a collector might go out and make 100. Some of our top collectors would make 400 specimen collections during one of our online events. And even if you're trying to mail those, just the bulk of those is quite substantial. And we're really not looking to describe all of these species at this point. Our goal is to document the biodiversity that's out there at this point. And that's another philosophical point, I guess, is to me, it doesn't really matter how long it stays Russula species Indiana 147, because I'm still able to link the collections together. I'm able to have a morphological description of what Russula species 147 is, because we probably have a dozen collections of Russula species Indiana 147. And you can pull them up on INAT, link all the pictures together. You can see when it occurs. You get a pretty good seasonality profile without a name. You can get a really good morphological description without a name. You can get a range without a name, just by linking all of these sequences together. I refer to it as linking like with like. And so that's really what our underlying goal is. And even if you were to want to describe this Russula in the future, you don't really need a bunch of different specimens to do it. You don't really need much more than just a portion of the cap. And in this case, I'll often tell people with Russulas, it's totally fine. If there's a large Russula, I don't even want the whole specimen. I want like maybe a quarter of the cap, or for a really large one, I might want 1/10 of the cap. I want a bit of the spore bearing surface, but also take good images to go along with that. And so I would say most of our collections are not the traditional, these large bagged collections, just due to space and time constraints. And if you're, imagine if you're trying to dry out 100 specimens, you're just somebody that's working at home, just the amount of dehydrator space it would take if you were making large collections. And so just functionally, saving specimens in the way herbaria have historically wanted them is not something we do. I've also had herbaria tell me before, literally I had one of the top herbarium people in the country is tell me, why are you sending me a single mycena? And to me, in my mind, it was almost like, how could you even ask that question? There's many mycena that just grow singly, right? You're not always able to have a large collection of these things, and a small single dried specimen might be all that you get. And if you use the traditional method of, that most herbaria managers would prefer, I would say, as to not collect those types of things unless you have, like a larger collection that's going to be really useful for time, throughout time. That's really what they want to see. But you have to think about it from their perspective in this case. They're taking on the cost and the time of storing these things in perpetuity. And so they want to have the most efficient use of space in their institution as possible. But I would say even a portion of a specimen saved is highly valuable if it has a DNA barcode, because you're able to link it to every other collection of that species that's ever been made. And so when you're working the way we do, any one single collection is not very important. And so we if we lose a specimen, it's not that big of a deal. If we only have a portion of a specimen, it's not that big of a deal, just because we're able to link it up through DNA to every other collection that's ever been made of that species. And I guess that's I've been going on there for a bit. But no, that's a long way of saying no, we don't actually save the whole specimen of each time. And me personally, if I'm walking through the woods and I see a Russula that's been eaten by a squirrel, that only has half of a cap sitting there, but it looks interesting for whatever reason, I'll still save that and dry it. And that's fundamentally different than most people would consider doing. I would say. But our DNA sequencing is cheap enough that we're limited by the number of specimens and not by the cost or time of it. And so even lower quality specimens, I'll sometimes save, and we got lots and lots of new species from low quality specimens, because you don't get to choose, the quality of an interesting specimen when you find it, and it might be a very long time before you find it again. And so, but being able to document that this species occurred at this place in this location at this time is really to me the more important aspect as to whether or not the specimen's in idealized condition. And so the crossed off species were misidentified. This is another question. Going back to that agaricus slide. So the crossed off species were misidentified, to put it in layman's terms. Well, yes and no. So I guess I would say a lot of those were actually misidentified. And by misidentified, that would mean more along the lines of they had been described previously, and maybe the person applying that name had no idea that there were other names in play, or maybe it was just kind of a field identification that was put into an herbarium. But something like Agaricus arvensis, that top one, supposedly it might occur in Indiana, but we have no evidence for it. Basically, everything that we had been calling Agaricus arvensis is actually right here, Agaricus crocodilinus in Indiana. And so we have no evidence for arvensis, and everything that we'd been previously utilizing that name for many collections, but that name, we no longer use that name for any Indiana collections. The same goes with Agaricus campestris. It's 4 or 5 different species now that look somewhat similar, and so we don't actually use the name Agaricus campestris for anything in our state anymore. How easy is it to see the differences on DNA sequence between specimens? We'll talk about that in a second. So I'm going to switch gears here in a moment. Let me go ahead and proceed. It's easy for me to get sidetracked with additional commentary. And so how can we document the most biodiversity, the broadest scale, shortest time frame, lowest cost, highest accuracy? This is what we've ultimately come to, is that there really are no other options that are viable to try to really achieve all of these things and to balance all of these competing goals. The collaborations are to get lots of specimens, and the DNA barcoding is to be able to link like with like. So I mentioned we're going to switch gears a little bit. We have a quick sip. So what actually is DNA sequencing? Let's kind of talk in real general terms, kind of from the beginning of what this even means. And so if you go take a biology 101 class, you'll learn that DNA molecules are made up of 4 base molecules known as nucleotides. So there's A's, G's, C's, and T's. A fungal genome has 50 to 100 million of these A's, G's, C's, and T's that consist of their genome. And that's what we refer to as a genome. And all DNA sequencing means is using some type of technology or process to determine the order of those A's, G's, and C's, and T's within the genome of an organism. And so DNA sequencing, that's all it means, is to determine the exact order of these A's, G's, C's, and T's within the genome of an organism. And that's different from DNA barcoding, because as I mentioned, a genome for a fungus might be 50 or 100 million base pairs, 50 to 100 million A's, G's, C's, and T's in some order. DNA barcoding essentially takes a small slice of that total genome, something on the order of 600 to 700 A's, G's, C's, and T's, and looks at that very small region known as a barcode region that's been selected specifically because of its ability to identify an organism. But DNA barcoding is different because it only looks at a small section of that really long genome in order to make the assessments of whether or not these things are identical or different. And we most commonly use a region called the internal transcribed spacer region, or the ITS region, if you're into DNA of fungi at all, you probably heard that term. And that's all it is a 600 to 700 base pair region of the genome that's been specifically selected in order to aid with identification of species. There's a bunch of other regions that people utilize for fungi, the large subunit, small subunit, RPB2, EF1, and others. But ITS is the most commonly used for a number of reasons I'm not going to get into today. So when you're thinking about, the obvious question should be, well, if a genome 50 million base pairs and you're only looking at 600, 700, what can that barcode tell you? And so my initial answer to that is almost nothing, but also quite a bit at the same time. And so if we were to look at, let's see, if you were to look at, say you take one of those DNA barcodes, those 600 A's, G's, C's, and T's in some order, if you look at that in isolation, if you look at that alone, it's not going to give you basically any information. Nobody can interpret or get any meaningful information out of any one DNA barcode, those 600 A's, G's, C's, and T's. You can't tell what species it is, what genus it is. You can't tell whether it's a basidiomycete or an ascomycete by that sequence alone. The only way that you can start to get information from DNA barcodes is to compare it to some type of reference or to another other DNA barcodes from other specimens. And that's really where it starts to tell you quite a bit. So any one DNA barcode in isolation tells you essentially nothing. But that DNA barcode can tell you quite a bit in the context of other specimens that have also had DNA barcodes. And these other specimens are referred to as reference data. And that's where it becomes really important to have lots of reference data from other species to be able to ask all the questions that scientists want to ask in all of these different areas where DNA research is helpful. And so that's one key thing to keep in mind is this reference data, and the quality of this reference data is very, very important to be able to make use of any DNA barcode data at all. And so if we look at an example, over on the right, this is an observation from iNaturalist. And if you look over to the right, we include our DNA barcode. That's that 600 ordered set of A's, G's, C's, and T's. We include that on our iNaturalist observations in the observational field section. And how do you begin to make use of that data? Well, typically the first step is to run what's called a blast search. And if you look at the bottom, those are some results from a blast search. Blast is a piece of software. It's an algorithm that was developed and implemented by I'm not sure who developed it actually, but it's been implemented by the US government, and it's seen as kind of a central repository, a warehouse for DNA data. And that's usually the first thing we turn to when we're looking to make use of our barcodes, is we take that DNA barcode and enter it into the blast search to get our results. And you can think of a blast search as almost like a Google search for DNA. So it takes that long chain of DNA, those A's, G's, C's, and T's, and give you the results that are most similar to it, and returns them in almost a Google-like format. And so if we were to expand this a little bit, and I'm not going to go into this in much detail, just because interpreting blast results, and the question a second ago was, how easy is it to see the differences on DNA between specimens? That's something that's, I could do an 8-day course on that. The answer to that question becomes very, very complicated very, very quickly. I mentioned early on at the beginning of this talk that it's not always conclusive. It is often somewhat subjective for a bunch of different reasons, but just even the interpretation of these blast results can be quite subjective. The way you interpret this, just briefly, is there's a percent identity column over here to the right. And the first two entries are 100% identity, then drops down to 99.85, drops down to 99.84, and then drops down to 95% similar. So what this is essentially telling you is these top two DNA sequences, they're both mine, by the way, match it at 100%, they're identical. Then you have another subset of specimens that almost match it, and 99.85, 99.84% similar. And then I have this thin red line here because that's where the species should likely be delimited, because you see that big drop in identity. So it goes from 99.84 to 95.39% similar. And so this is a pretty obvious place that the species should be delimited. It's not always this obvious. What if it dropped from 99.84 to 99.3? You know, there are many scenarios to where interpreting what your species is based on the reference data quickly becomes difficult, just even in that context. But then if you look at it as a secondary context where these red arrows are over to the left, even though we believe everything above that thin red line is probably going to be the same species, I see 1, 2, 3, 3 different names in play, 4 different names in play. We have Validipes, Underwoodii, Flavadellis. So there's been specimens that have been identified under all these names. And I can tell you, if you look on mushroomexpert.com, they don't use any of those. Quo has it as Liquidiae. And 1, 2, 3, or 4 of these could be misidentified. They could all be misidentified. You know, everything, all of the names you see listed here could be misidentified. And there could be another name that just hasn't been uploaded to public repositories yet that could be in play for all of this. And you never know until the type specimens have been sequenced. And so another reason why all of this is kind of subjective is you're essentially relying on the reference data that other people at some point in history have put into these public repositories. And the reference data might conflict with each other. It might be unclear. It could just be outright wrong. And you really have no way of knowing that. And the only way you can begin to make that determination is whether or not a type specimen has been sequenced. And so if you don't know what a type specimen, whenever a mushroom is first described, and so whenever somebody makes a determination that says the species is new to science, they create a formal species description for that species, and they designate a specimen, a physical specimen that's put in a herbarium somewhere that serves as the forever designation of what that species is supposed to be. And so along with every new species that's described, there's also a type specimen that's actually in an herbarium somewhere, most typically. And one of the issues is, when we were talking about the reference data that's available, the only way to get a ironclad, and even then it's not always ironclad, because remember, we're still kind of determining in probabilities here. You know, it's 99.84% similar to a type specimen. But in a general sense, these type specimens do give you a, you know, quasi-definitive understanding of what the DNA barcodes of these species should look like. ITS doesn't work for everything. We mentioned it's only 600, 700 base pairs out of the 50 million. But it works for the vast majority of things. I usually say 95% or more of species that are out there can be easily resolved by ITS alone without the need for any additional genes. But there are that 5% when you're dealing with, you know, 20,000 species in North America or more. That 5% is quite a substantive number that do need additional barcodes. But the vast majority of the things that you collect and the vast majority of the things you look at, that one DNA barcode of that one region is going to be sufficient. A couple more questions in the box, just briefly. If the DNA of the fungi to look for in the search cannot be found, is this a new species? And the answer to that is no. And so that's obviously another big problem. Is so let's say that all that means is if no matches come up in your Google search for DNA, all that means is that no one else has ever uploaded it before. That species has not been DNA sequenced before. There's still the majority of the species worldwide, I would say, that have not received a DNA barcode, that have not been DNA sequenced. And so absence of evidence does not equal whatever that saying is. Just because you don't have a matching piece of reference data within this public repository does not mean that the species is new. It means, but that does tell you that more work needs to be done there, and that the DNA barcode that you generated is particularly useful because now it associates that barcode forever in perpetuity with pictures, with images. You now have a face to what that DNA barcode is, and that helps people in all kinds of different ways. I mentioned the environmental DNA. Just briefly, I'm going to burn through my time here pretty quick, but if you think about environmental DNA that we talked about, there's a lot of fungal ecological researchers that are looking at the DNA of mushrooms and fungi in the environment, but all they get back are these DNA barcodes that don't tell them anything about what this thing looks like. And that's why these DNA barcodes that are generated from specimens, especially ones with nice color images, are so important, is because they help fungal ecologists put faces to all of these DNA barcodes that they're generating from environmental DNA. And so I'm going to need to step it up a bit. I'm approaching time here, but just another quick example. There's a problem within the scientific literature that's not often discussed. And if you were to look at this 96 North American Hebeloma Revisited paper, the problem of synonyms, this one species has been described 25 different times under 25 different names throughout history. And so when you're looking at this list, it's possible that Validipes, Underwoodii, Flavidelis, Lacourtier, it's possible that they're all the same species, that it's only one species and they're all synonyms because this species has been described multiple times throughout history. Alexander Smith described Hebeloma mesophaeum 20 times in a single book or paper. Veiled species of Hebeloma in the Western United States. He described that single species 20 different times, one author. But this species overall has been found now. There's 25 different names that have been in play for this. And so that's just another quick example. Even for seemingly distinctive species like Rhodotus palmatus, this has been described 1, 2, 3, 4, 5, 6, 7 different times throughout history. This will ultimately be renamed Rhodotus reticeps. And that's something I'm working on as kind of a side project as well. But even these common species have been described 7 different times throughout history. And so the best reference data is from the original specimen is from the type specimen, and that's the specimen that the description of the species is based on. And very few of these specimens, type specimens, have been sequenced to this point. That's actually something I'm going to be working on to quite a substantive degree here starting this month, actually. Not going to talk much about that. Let me try to get to nanopore to wrap this talk up a little bit. I would mention that we started out with a type of sequencing known as Sanger sequencing. It was developed in the 1970s and it's thought of as kind of a first generation sequencing technology. This cost us about $5 to $10 per specimen. It required us to send samples out to a commercial lab. And we now use a newer process called nanopore sequencing. And so you can see a picture of the actual unit on the right, and it's very small. It's a handheld unit. You set it on a lab bench and plug it into a laptop. The unit itself costs about $1,000. The add-in that you need for barcoding costs about another $1,400. So you have about $2,500 in the equipment itself. We only started it this year. I bought the unit, I think, in May. We had our first results, I want to say July. And so it's a really new thing for us. And to this point, I really don't know of anyone else using it for barcoding of fungi. We delayed implementation of this for a number of years because one of the drawbacks of nanopore is that it has a higher error rate than Sanger sequencing or even Illumina sequencing, if you've heard of that. And that's one of the reasons why we didn't adopt it, because we had always been told that this error rate essentially means you aren't going to be able to get accurate and reliable barcodes. But the way this works is, I'm going to skip that. I guess I will not go through too much about the error rate other than to say the recent product releases are significantly improving the error rate. So every generation of this device, and they just released a new one last month, a new flow cell. That's the unit on the top, that black portion on the top of the device. That's called a flow cell. And that's what they iterate. That's what they improve continuously. And they just put out a new one here this last month that should significantly help to decrease the error rate. But this is something that's going to be continually improving over the course of time. What I can tell you about the error rate, my results are Sanger compatible with the previous generation of flow cell. So the previous generation was they call them 9.4 flow cells. And the results that you can get from that flow cell are easily Sanger comparable and work well, very well, just as good as Sanger sequence does in terms of barcoding. And so I was actually surprised by this because it was something that we had been delaying for so long because of this apparently unwarranted fear of the error rate. But especially with the latest release that just came out this last month, that error rate continues to improve. I haven't been able to run a new flow cell yet, so I don't have data on the latest error rate, but it was already Sanger comparable in terms of the quality of the output barcodes with the last generation. Some of the advantages of nanopore sequencing, the cost has dropped about 10x. So we're looking at about 60 cents per specimen. Most of the cost of that goes into the extraction and amplification parts of the process and not the actual sequencing. And so it's possible that you could drive that price per specimen down even more. There's a significant time savings. And I'm going through this kind of quickly without talking too much about it. And so for those of you that know about gel electrophoresis and sequence editing, you don't have to do either of those, which is, to me, a very important time-saving part of this process. And the sequencing can be done at home. You no longer have to mail off your specimens to some third-party lab and hope the results, A, hope your samples arrive intact, and B, hope the results come back well. If a sequencing run fails, which I've only had done once, you can just repeat it at home yourself, and it works. So I guess I would note there when I say it failed, I did have to use a new flow cell that costs about $100, but you're doing 1,000 specimens at a time. And so $100 is almost like a drop in the bucket compared to what we were used to be using for sending out specimens for Sanger sequencing for 1,000 specimens. There are some disadvantages to using nanopore for barcoding. You still need to amplify your DNA. You use lots of specimens in a run. It's more cost effective than Sanger at utilizing about 30 to 40 specimens, but we use 960 specimens for each run. You need a good computer, particularly a good GPU, a graphics processing unit, because the data analysis is very complex and it takes a lot of time. The software is Linux based, so you need to know Linux if you were to try this yourself. And the post-sequencing analysis is more difficult. There is a pipeline that kind of simplifies it, but it does use 3 or 4 programs in a Linux environment. The one question is, do you need to do anything other than PCR before actually doing the sequencing? So the overall process is you do the DNA extraction. So you break open the cells to release the DNA into your solution. You do DNA amplification, just as you would with Sanger. So you turn a little bit of DNA into a lot of DNA. And then, one second. You have to do a chemical reaction that ligates this little motor protein onto your DNA. And there's a couple other smaller things, but fundamentally, the entire process from amplified product, from PCR product to sequencing, that entire process takes about 3 hours, with about 90% of it just waiting for reactions to occur. And so essentially all you're doing, other than to do the actual sequencing reactions, is doing 2 very basic chemical reactions. You're just mixing 3 chemicals together and waiting twice, essentially, and doing another chemical reaction that cleans it up, a bead cleanup, if you're familiar with that. And so the overall process post amplification takes about 3 hours, with about 90% of it waiting. And so there's not much else other than DNA amplification that you need to do. And overall, it's about, overall, I say it's 100x efficiency gain versus Sanger. You get about 10 times more throughput because you're doing about 10 times less labor and work throughout the entire process. And you have about 10 times less cost And so I often refer to it as a 100x efficiency gain for our projects. And that's what's really allowed us to almost do in a year what took us, 5 years previously. We've been able to do, I think we're up to 7,500 specimens now, finishing up another 1,000. So we'll be about 8,500 specimens this month that we've done since July. And to do 10,000 specimens took us more than 5 years previously. And so just the amount that we're able to process with this newer protocol combined with the lower cost means we're no longer limited by time or cost in terms of our DNA barcoding effort. Now the primary limiting factor is just the number of specimens that we have available. And that's one of the reasons why I really put a focus on those online forays right up front, because if you're really trying to make the most use of this type of technology, you really need to have a continuous pipeline of new specimens that are continuously being fed into your process. That way you don't just have a lot of dead time and unused materials, essentially. Because these flow cells, the pieces of equipment that do the actual nanopore sequencing, they could actually do 2, 3, 5,000 specimens on a single flow cell that costs $100. And so you could actually, if I had the ability to have many, many more specimens, we could easily scale it up even further and do more than 1,000 at a time. But the 1,000 mark does it nicely for us. It allows us to, so if you think about it, if you were to do much more than 1,000, how long would it be between the time somebody collects something and you are actually giving them the results? We could go up to 5,000 specimens on a run probably, but then people aren't going to have the results for, a year. And that's not ideal. We want to return our results to people as soon after as an event. And so those kind of factors as well, other than the ultimate capacity limits of this type of technology are what cause us to kind of limit it to 1,000 specimens at a time, but you could go a lot higher than that. And so with that, I would be happy to take any questions if anyone has any. I know I'm a little over time, and I would I'll stick around for as long as you have questions that you'd like to ask. I thank you guys very much for having me. And to me, this is a really exciting technology for us. It's really kind of changed the game for us in terms of our capacity and our ability to document biodiversity. And I would really encourage, I guess, if there was one takeaway that I would ask for everybody that's listening today, is to start collecting specimens and saving specimens, and to make that just a continuous part of your processes out in the woods. Because even if you can't afford it today, or even if your club doesn't have a DNA sequencing program today, starting to build that biodiversity database is going to be really important, because whenever you guys do start, or at some point in the future, someone's going to come and say, hey, do you guys have any specimens from there? If you have 1,000 specimens, let me have them. That's not going to be too uncommon in the future, I would say, is people are just going to kind of be scrounging for specimens wherever they could get to try to document this biodiversity. I'm already kind of tapped out on what I can do for Indiana, and we have a substantive pipeline of new specimens coming in regularly. And so I'm actually starting to go into herbaria now, just because we need more specimens. That's our limiting factor. It's the number of specimens that are available. So if there's one thing to take away from this, just the value, the long-term value. Even if you don't have the ability to do sequencing today, tomorrow somebody will be making use of your specimens, I would guarantee. And so
Steve,
thank you for having me.
Yeah, your enthusiasm is infectious. And you often in your talk spoke of the term we. How big is your team, and how do you handle your specimens when they arrive? Because if you're receiving 3,000 specimens in a year, there's some logistics that are involved in this that could help you handle it. So what is the we in the matter, and how do you organize to handle the specimens and get them into the
So yeah, when I say we, I'm primarily referring to me plus collectors. So in terms of the back end, there's nobody else that's really helping me with the sequencing. And for most of the time, it's just me kind of doing specimen triage as well. And so when I say we, it's more or less me doing the back end and then the collectors that are out collecting. And obviously I'd like to spend more time out collecting. But this is the back end work to me is kind of what's necessary to make something like this happen. And so trying, I guess, as a club, it's not really something you would want to try to do as a committee. You are really going to need like 1 or 2 people that are passionate about, getting this type of data. Just because the amount of organization that's required, you really kind of want to standardize the processes, and it's easier with fewer hands in the bucket, I would say. And so what, I guess.
So the role for members in the society then would be to supply the specimens.
Yeah, that's kind of the limiting factor, is just being able to cover as much land area as possible and to take as many pictures as possible. And then, I mean, even on their end, it takes it's a long-term training type thing, because they have to be able to produce specimens that you can use. And so they have to be organized and structured to where once you get a few hundred specimens at the sort, for whoever's doing the sorting, they have to be able to link them all back to like iNaturalist observations. And so even from the collector side, it's not like it's the easier portion by any means, because just the amount of time that it takes to organize all your iNaturalist observations, to annotate all of the specimen bags, to dry them out without getting them disorganized is quite substantive. And so I definitely wouldn't, I would never try to make light of the amount of work that's involved. I mean, it's a very substantive amount of work. But in terms of like large scale collectors, we probably have about a dozen people or so that, produce hundreds of specimens a year. Then we have a larger universe of smaller collectors, right? That might send in 10 or 20, which is more rational for most people. I don't know if you've ever gone out in a day and collected 100 specimens and then actually organized them with the images and drying, and that you're going to spend several times as if you spend an hour in the woods, you're going to spend several hours just organizing and annotating and uploading and databasing, just from the collector side.
Yeah. Martin has a question.
Yep.
Yeah, thanks, Karen. So are you using iNaturalist obviously for the photographs, but there's all kinds of other characteristics, smells, color changes, bruising, and stuff like that I think is, just important morphological characters that sometimes you don't see in photographs.
Yep.
So basically, all of those have to be well noted in your iNaturalist.
So that comes down to the quality of the collectors, right? So that's part of the training process over the course of time. And so I would say most often they don't annotate those things, but they will take pictures of those things. So, let's say for a bolete that there's a reaction for ammonia, right? They might put the ammonia bottle in the picture next to the specimen and take a picture of the color change. Obviously, you can't do that with smell. But we really try to encourage photographing, because it's less subjective, I would say. Photographing as many of those features as possible, but then for something like smell, just toss it in the iNat notes. Yeah. And that's part of specimen, part of collector training, is to, once people start to really understand what are the key features that are important for a lot of these groups, that's when they start to improve their overall documentation ability. But no, we don't try to impose so something that is never going to work is like if you have some long list of things that have to be filled out for every specimen. Nobody wants to do that because it's not fun, right? These people are donating their time essentially. And even without that, you have to remember, let's say they gave no documentation. Let's say they gave no photos. and they just send in a specimen, it's still very valuable because you only need one specimen where someone's noted the smell at some point throughout history that you can now link that, this lower quality information to. Essentially, you really only need one documentation of it throughout history. You don't need everybody smelling every sample. You need, if you have 10 collections that are now linked together by DNA, all with pictures, you only need one of those collectors at one of those points of time to have noted the smell to know what it smells like. Right? So it's almost a waste of time to even try to encourage that for every specimen that comes in for everything. And I would also say, I guess, my philosophy is a little bit different there as well, because let's say we have 10 specimens that don't annotate the smell, and then I want to describe it. Well, all I have to do now is say, hey, post on our Facebook group, this specimen has been found at these locations during this time of the year. Someone find it and send me the specimen and tell me what it smells like. And every time I've ever done that, it's worked, right? And so it's kind of a different philosophy. It's almost like backing in to the information that you're missing, rather than trying to proactively collect all of this information up front, which nobody's going to do anyway. And that takes all the fun out of it. It's already tedious enough to have to, do all these uploads, to do all this organization of dried specimens. Man, if you can get them to do that, that's more than enough for me. The last thing I'm caring about is smell.
Right. So are you interested in moving to Alberta and get our stuff? Because obviously, you need somebody who's going to play your role. And it seems that that's fairly significant.
Yeah. So I guess my role is for a local club. I would say you don't need to do most of what I do in terms of the sequencing. So I do offer sequencing, at a very low cost, but there's going to be, I would suspect anyway, there's going to be a lot more people that are offering it at a very low cost. I would say for most clubs, it would make 0 cents to actually start a sequencing project yourself, to where you actually go buy all this equipment, start your own lab, just because the cost of it. So like, just as an example, for 1,000 specimens, I'm offering it for $1.75 currently. So that's $1,700 for 1,000 specimens. Where in Sanger, that would cost like $10,000. And so the cost of this is only going to keep going in the negative direction. And so doing everything that I do probably just isn't the most cost-effective thing or time-effective thing for most clubs to do. The main thing I would say is starting that structure of being able to get specimens, from lots of people through these kind of large scale citizen science collaborations, and to start getting the systems in place to get people trained for how to collect, what type of, how to take their pictures, what type of information is important for which types of groups to document, and getting the process, the workflows in place for specimen processing. If you can just do that part, I think there's going to be lots of people in the future that are able to effectively, very, very cost effectively process the actual sequencing components for these.
So sorry, for that $1,700 on 1,000 specimens, does that include doing the blasts and all of that?
Yeah. So DNA extraction, amplification, sequencing, post-analysis, having your own dashboard essentially for all your results, having all the sequences organized and having names applied to all of the specimens and uploaded to GenBank.
Well, there you go.
So basically the whole process.
So you're saying you can do that whole process for how much per specimen currently?
$1.75 at 1,000.
Yeah, it certainly would make a lot more sense for us to send it off than to try and get our own PCR equipment and whatnot. Yeah.
Yeah, I mean, it's but that also just simplifies the whole process too. If you don't have to have somebody, because that's a whole different skill set than someone who's able to organize specimens, having somebody with lab skills. and that's also interested in, doing thousands, that's kind of a whole different person than it would take for somebody to just organize specimens and to start getting your biodiversity database, your biodiversity bank of specimens in place.
One other question. So if we have one specimen, or like we have our herbarium specimen, how much of that specimen do you need for to do the work where we can still retain the specimen? I assume we're going to lose the specimen once we send it to you.
No, so what I typically refer to as a split. So a split of the specimen, and I do request anything that I do that I actually have a split sent to me. But you can retain the majority of the specimen. The amount that's actually used for this process is, so for Sanger, I would typically say about the size of a grain of rice. And for this, I would say probably about a quarter of the size of a grain of rice is all the amount of tissue that you need. And so when I say split, I would really only need like, a piece smaller than a, I'm trying to think of I was going to say a dime, but you guys are from Canada, so you probably know what a dime looks like anyway. But like half of a dime or something like that is a typical size for a split. Yeah, just like a very small portion of a specimen. I wouldn't need the whole specimen. Most of the specimen can be retained locally, and I wouldn't even want the whole specimen because that's more that I have to deal with, right? So I would really only need just a very small portion of each specimen.
And what would you do for smaller macrofungi, quote unquote macrofungi, who just exist in very small fruiting bodies?
So as long as you can see it, it's going to be enough tissue.
Okay.
So if you can visibly see it with your eyes, and oftentimes, so like the thing that grows on trichoptum, the Phaeocalicium polyporaceum or something like that. It's just these little tiny fairy wands that grow on the top. But I couldn't even see them, right? And so I just kind of scraped the cap a little bit with my tweezers, with my forceps into a tube. I saw nothing in the tube, but it came back with the sequence. And so I guess you don't even need to actually have enough to see. I just knew it was there based on the picture. Like literally what I did, I pulled up on iNaturalist a picture of where these things were on the specimen, and then just scraped that area into a tube, and it came back with the target.
And do we need to worry? Oh, sorry, go ahead.
Yeah, I was just going to say, the amount of tissue you actually need for this process is very, very small.
Do you need to worry about getting like just the interior of the stipe or stuff like that, just for concerns about contamination on the surface, or the stuff kind of evolved to the point where that's not necessary?
Yeah, so that's something that I didn't get into as a benefit of nanopore, is there are no concerns about contamination. So let's say you get your target and your non-target. What it does is essentially provides both sequences from both specimens. So let's say you have aborted inuloma as an example. It's going to give you both the aborted inuloma sequence and the armillaria sequence within the same result, as two separate sequences. So the same thing with like hypomyces on the bolete. You'll get the bolete sequence as well as the hypomyces sequence, both associated with the same specimen. So essentially, it doesn't matter if there's 100 different fungi involved. You can just do the one extraction, the one PCR, and it's going to disaggregate all of those various types of fungi that are involved in the sample. And so contamination is not a concern.
That is amazing. Awesome.
So as opposed to Sanger, if you were to have hypomyces on the bolete with Sanger, you would send it in and your sequence would come back all muddled, and you wouldn't be able to disaggregate the two reads because they're just kind of compiled on top of each other. But with this, you actually get two independent sequences.
Do you also do soil blasts?
Also do what? I'm sorry?
Soil blasts? Like if we sent in the soil sample?
I could, but that's not. So that's actually probably going to be one of our next steps in Indiana, is to have a year of people out collecting soil. The problem is I need a better pipeline to process it, just because it's more difficult than working with tissue. The steps that you take for each sample take longer, and so I need to find kind of almost like a quick and dirty soil extraction method. And typically the thing that's most available right now are these kits that are expensive. They're like $15 a sample, and I can't do hundreds of samples at $15 a sample. And so kind of improving the current protocols on the soil extractions and amplifications are probably going to be a next step for me.
Well, I hope I can speak for everyone. I think you are an inspiration for our society. I don't know if you could sort of gather by the way that the conversation has been going, we kind of are on the cusp of developing a good storage system, and just deciding what to do with our specimens and how we can.
If I could, let me make one more note. Someone asked what I do with the specimens.
It's going to show us what he does with the specimens.
Can
There
you
they are.
see?
Yeah.
So it's just a bunch of file cabinets right now. Within them, I don't know if this will go all the way over.
So Ziploc bags and
Ziploc bags within larger Ziploc bags. In a file cabinet is essentially the story. And so that's not a permanent solution, right? That's kind of a temporary solution until they go off to herbaria. But that's how we store them all right now. I have people send in the specimens in a Ziploc bag, write the iNaturalist number on the exterior of the bag, and that allows me to quickly get the metadata that I need to process a specimen.
Awesome.
And then, yeah, I just store them in this large bank of file cabinets behind me for the time being.
So you've streamlined the organizational end of it, but you focused on the analysis of the.
And I do have a paper. There's on the online forays. If you search my name in like online forays, you'll probably find that paper. It's a preprint. I haven't published it yet anywhere, but that kind of talks about a lot of the processes that we use for streamlining those online forays and specimen collection and triage and all of that.
It does make it less cumbersome and less worrisome for someone who's very, someone who perseverates. And if you just have to do a few things, then it most likely will be done and shipped over to wherever we're collecting.
So
yes.
that's one of the things too. You have, in the past, people have always tried to get these long collector sheets or something with each specimen, with all of this data, saying what's the width of the cap and what's the taste and smell. But the more hurdles that you put in front of people, the less number of people that are going to participate, and the less actual data you're going to be able to get back in. And so really trying to simplify the process as much as possible. There's always going to be people that go above and beyond. And I would much rather have more people participate at a lower barrier to entry, and then have those people that are going to go beyond anyway, go ahead and do it. Right.
Yes.
But then getting more people with that really, really base level barrier of entry has really been beneficial, because those are the people that you want to get started in this process, because eventually they become the people that go above and beyond, and start taking, notice their progression of pictures from year to year, right? Yeah. They're like, crap, I need to get a better camera. And then, the next year you see a substantive improvement in their pictures, or that, they start seeing how other people do things. They're seen as more experienced people, and, they start taking on additional roles and functions. And so it's a process, right? And so you want to make the barrier to entry as low as possible to begin with, because that's how you develop your pipeline of people that are become your model collectors, several years from now.
Do you have forays where you have your star collectors, your model collectors? Because there is a lot of good feeling and sense of community that goes with a larger group foraging together.
Yeah, so we do during our online events, we actually will have physical events throughout that entire thing. So I think for that summer foray I mentioned, we had, I think, 10 different forays across the state during that week.
Yes.
And so, I'll have all my top collectors essentially, they want people to come help them spot mushrooms, right? So they want more people to come visit them. And so I just say, give me a date and time that you want to be out. And we schedule a foray with them on that day, wherever they are in the state.
Wherever. As I said, I can't say it enough. It is an inspiration, and it does help clear our thoughts on the matter. And we meet once a month, and our meeting is upcoming. So while we might be like mycelium, doing our work quietly underground, and we might pop up again in your life. So I really thank you for coming out. I mean, coming on Zoom is not trivial. So I thank you, and the check will be in the mail.
Okay. Yeah, I thank you guys. So to me, this is almost like a privilege to be able to do this type of thing, just because I don't know why personally, it's like an irrational goal, you know, especially starting with
Yes.
the Mycoflora project and everything like that. It's an irrational goal for me to document all of the mushrooms that exist, and not just in Indiana, but like everywhere. And so for me, you know, helping other people to understand what we're doing in Indiana, and maybe helping them to, you know, streamline some of their own thoughts and processes wherever they are, kind of helps my broader goal. And so to me, it's almost like just an extension of, you know, what I enjoy doing. So.
Well, we're much in the same kind of idea here, than that we've been trying very hard to get good documentation of all the species in Alberta, but we have not yet been able to fully embrace the DNA work as well as you have, and I think it's going to really help us in our future planning and processes.
And yeah, feel free to ping me. Anyone listening, feel free to ping me whenever. steve@hoosiermushrooms.org. About whatever questions that you have, or, you know, thoughts that you have. Don't try to message me on iNaturalist or something like that, because I never read those. But yeah, definitely, like on Facebook, feel free to message me, or on or by email, because yeah, like I said, I'd love to help you guys get set up with whatever you're interested in doing that involves kind of mass documentation of fungi.
So I thank you again, and we will be in touch.
Okay, thank you all.
Thank you so much. It was excellent.